How much time does it take to get the vaccine? What's the process it goes through? So the screen is all yours, Professor Vanham, and he is going to talk to us on the roadmap towards vaccine against COVID-19. So Professor Vanham, the screen is yours and the audience is yours.
Thank you very much. It's really an honor and a pleasure to be with you from such a large distance. And I hope you can hear me clearly from my study room, because we are still living in a partial lockdown situation in Belgium. So I put together the present knowledge about vaccine in a nutshell, really. So I don't see how my screen goes down. That's it. Okay, so this is evidence from a Chinese study that people who are infected with SARS-CoV-2 after their admission to the hospital gradually develop, in the left upper panel, gradually develop neutralizing antibodies in their serum. And as you can see, it reaches a maximum after 1 to 2 weeks after their admission. And in the right upper screen, you can see that it remains stable when they revisit the hospital a few weeks later. Below, you see that the titer, in fact, is very different in different people, and that at least 10% of them or so does not have a clear neutralizing antibodies, and that about 30% has very low levels. So that asks already the question, will these people be protected against reinfection? Now, what are the key features, the salient points about the COVID-19 vaccine? And as you see, it's from a recent review. As you see, the first point is minimize undesired immunopotentiation. So why is that safety issue put first? Because during the development of SARS and MERS vaccines, of course, SARS and MERS are not present anymore in the human population, but efforts have been done in animal models to develop vaccines. And during these efforts, and also the development of other coronavirus vaccines, sometimes after vaccination and challenge, in fact, the pathology got worse because of a number of mechanisms, amongst which antibody-dependent enhancement. So safety is an important consideration. And then the two next considerations is like, what are the main target or the first priority target populations for a vaccine? Of course, healthcare workers, but also people of age. And that, of course, poses a problem on itself, because we know from influenza, from pneumococcus, that people above 60 have less good immune responses and are less protected. Anyhow, this is a very important slide from a recent review. As you can see, it shows the virus, it shows the spike protein, a trimer with the receptor binding domain, and it shows the 6 principles, platforms on which vaccines are being developed as we speak. And below, you see the classical types, which is inactivated virus or live attenuated vaccines. I will show you an example of the inactivated virus. Of course, if these principles would work, it would be relatively simple and straightforward. There is a lot of production capacity in the world, and then we could have a vaccine in a matter of a year or so. But of course, it's not sure on beforehand that this will work. And so there are 4 other platforms. One is the DNA vaccine, the plasmid vaccine, which is a classical method to produce also the recombinant protein. But the DNA, the plasmid itself, of course, could be used as a vaccine and has been successfully used in animal models of other infections, but has never been successfully used yet in humans. Nevertheless, DNA vaccine could be used in a prime boost setup, for instance. Now, from DNA, of course, you can easily in vitro transcribe to messenger RNA, and there are at least 2 vaccines already in phase 1 in humans, based on the messenger RNA for the spike protein. Now, a plasmid DNA could also be inserted in a viral vector. And there are quite a number of companies and academic centers that use different viral vectors to produce a candidate vaccine. And finally, of course, we could also produce recombinant proteins for probably a prime boost setup. In the next slide, I will not go through all of it, and you can read it in the paper. The advantage and drawbacks of these different types are listed. And of course, as I already said, RNA and DNA vaccines have not yet been developed for humans, so that would be new and take more time. Recombinant proteins, we have of course experience with very good vaccines for hepatitis B and for papillomavirus. And with regard to viral vectors, of course, a lot of animal models exist. A lot of vaccines have been developed for animal models with viral vectors, but there is only one in clinical use, and that is the Ebola vaccine based on the vesicular stomatitis virus. Now, the rest of my time, I will show you 2 examples of vaccines that are in development as we speak. The first is the simple, if you want, inactivated virus. You see the production scheme here that is being developed in China at the moment. So you just cultivate the virus in large quantities, you inactivate it with beta-propiolactone, and then you go into a production process where you also add purification, etc., and you add aluminium hydroxide, which of course is a very classical adjuvant. So this is really straightforward and simple, and indeed it works in the sense that in mice and in rats you induce high titers of neutralizing antibodies. And then the next phase has been non-human primates, where 2 doses were used, a 3 microgram and a 6 microgram dose in a low number of individuals, I think 3 or 4 per group. You have also placebo, a sham, etc. So you see that the red dots is the high dose, induces neutralizing antibodies, and then upon infection you see the antibodies rise, but also of course in the non-vaccinated animals neutralizing antibodies appear. But if you look at the virus then in the throat and in the anal swab, well, the non-vaccinated animals of course keep a high viral load, whereas those that have been vaccinated with either the low or the high dose, well, there the viral load decreases. And in fact, the animals clinically, the animals with a high dose, the 6 micrograms, were completely protected against disease. So this vaccine is now moving towards clinical trial in human. So the next example is not really a SARS-CoV-2, but it's MERS that has been published, but it's an Oxford group with a shim adenovirus vector construct. But they say that in a press release that they have a very similar vaccine for cough already ready for use. So this is it's just published last week. This is the animal primate study where they use either prime with the vector, so 1 injection, or prime boost, 2 injections with the vector containing the MERS spike protein. And indeed, you see that antibodies and neutralizing antibodies are clearly induced. And then they did a challenge in these primates. So at the right side, you see the animals that have not been vaccinated. So the upper panel shows the lungs. You can immediately see that the lungs are heavily damaged by the disease, that there is a lot of infiltration in the middle panel, and that the brown dot here is the virus that is present in the lung. When they only give one shot of the adenovirus, there is less damage in the lung, there is less infiltration, there is less virus. And when they give the 2' boost, then the lungs are completely clear. There is no disease, there is no infiltration, there is no virus in the lung. So this is, according to their observations, a completely protective regimen with the chimp adenovirus construct. So to end up, I will return to the first, the first slide, the summarizing slide. So I gave you some examples of what is being developed, but in total there are at least 100 different candidates in the running, and I understand that there are also candidates in India, and I think we will hear about it soon. But of course, the animal studies is one, the first stage. Then you have the clinical stage, phase 1 stage, which is now entering, several candidates are entering phase 1. That's for toxicity, for safety. Phase 2 is for immunogenicity. That will take more healthy volunteers and several months. And then if everything goes well, you go to phase 3. But of course, that will be in real life. You need large groups of thousands of people probably followed up during several months, half a year, to be sure whether they get infected or not, whether there is a difference between the vaccinated and the placebo group. So as you can see, since most of these candidates are still either in the animal phase or entering phase 1, that it will be would be quite surprising that we have a result of a phase 3 trial before the end of the year. Most probably, if everything goes really fine, we will have the first results early next year with the first candidates. And then, of course, they have still to be approved by regulatory authorities. They will have to be produced in large amounts and distributed. So you can see that, if everything is really 100% fine, that there could be a vaccine available for priority groups, maybe in 1 year or in the autumn of 2021. I think that is the perspective in the most optimistic scenario. So I thank you for your attention. I hope you have heard my talk over such a large distance. Thank you very much.
Thank you so much, Professor Vanham. That was a beautiful overall perspective of how the vaccine is developed. Thank you so much. Sharing the dais from my side is Dr. Shashikala, who is the Director of the Basic Sciences and Translational Research at the AIG Hospital. They've been doing a lot of basic science work. So she's sharing the stage with me here. She is going to introduce Dr. Vikram, and after that Vikram will do his presentation, and subsequently we take the questions from the audience.
Good afternoon. I am glad to introduce Dr. Vikram Paradkar, Senior Vice President Technical Operations at Biological Evans, which is a biopharmaceutical company based in Hyderabad. Biological Evans is a very well known company in this part of the world, and it is specialized in the development of low cost vaccines and is a major supplier of these vaccines to international organizations such as WHO and UNICEF. We are proud of Biological Evans to have this. Dr. Vikram is responsible for process and product development in the vaccine area, and he is also responsible for clinical manufacturing of vaccines. So Dr. Vikram, welcome to the session, and over to you.
Okay. So thanks a lot, Dr. Shashikala, for your introduction. I'm actually coming from my office. We are part of the essential services, and so we are all working to produce vaccines. And I would be very happy to share how the vaccines will be developed, especially in Indian context, with some unique, considerations. Okay. As you know, Indian manufacturers supply almost 60% of the vaccine doses consumed all over the world. And more than 25 WHO pre-qualified vaccines are supplied out of India. We have 9 major manufacturers in India, and most are vertically integrated, which means we manufacture all our components, all the antigens, adjuvants, as well as the finished product. And several of these manufacturers have announced COVID-19 vaccine development, either through in-house work or through collaboration. These collaborations are with academic institutions globally. For example, Serum Institute is doing a collaboration with Oxford University.
Dr. Vikram, can I just interrupt you? Can you just make it full screen, Vikram, because the screen is not shared completely?
Okay.
Yeah, absolutely. Thank you. Thank you, Vikram.
Sorry. So Professor Vanham gave an example of the chimp adenovirus vector viral vaccine. That vaccine developed by Oxford University is now in collaboration with Serum Institute of India for manufacturing of this vaccine. We also work with vaccine consortia. So we also work with other industry partners to develop this vaccine, because our aim is to make sure these vaccines become available to large populations in the world. And Indian manufacturers have experience with all major platforms. Professor Vanham gave a very good introduction of all of those. I'm very happy to say that all of the manufacturers in India are working on all those platforms. And so we hope that a successful vaccine comes out of them, whether it be inactivated virus or a subunit vaccine or a DNA vaccine. I'm sure we'll develop it very soon. I just want to give you a brief idea about the pathway that is followed, especially under these unique circumstances where there is a tremendous demand for a successful vaccine, because without which I think we cannot fathom opening our society. So, we all start with what we call as discovery or proof of concept, where a vaccine is developed in terms of a overall combination of antigen and adjuvant, and it's demonstrate the immunogenicity in an animal model. Once that is done, we know now that this vaccine has a significant chance of becoming a successful candidate, and immediately start working on it to develop this. We develop the manufacturing process for this antigen. We develop a formulation which is with a suitable adjuvant, because not every adjuvant works with every type of antigen. So we have to select those and also demonstrate that we can produce this vaccine in a consistent manner. It is also stable vaccine. Once that happens, we quickly complete the basic animal toxicity studies. Most of vaccines, are based on existing knowledge, so we know what are the key pitfalls to avoid in safety of that, and so we already know what benchmarks to hit, and so we finally complete these toxicity studies with a proper matrix of dosages with adjuvants and so on, and we evaluate the maximum dose that potentially can go into human very quickly in animals. So we have very good labs here. We set up to do this testing, various species, rabbit, rat, mice, and with that we can confidently go into a phase 1 clinical trial. In these trials, we will test the maximum dose that we can envision. We also have to test the multiple regimen. Some vaccines are given as a 2 or 3 dose regimen, so those are also tested in this. And we are looking at safety first, but also very quickly immunogenicity, especially the neutralizing antibodies that are very, very important, because without this, the vaccine will not work in the target population. We believe for a product like COVID, if we demonstrate very good safety and immunogenicity in phase 1, we might as well get what is called as EUA, emergency use authorization, especially in frontline healthcare workers, frontline workers who work in other areas where they are highly at risk of contracting this infection. So if we demonstrate very good safety and immunogenicity in these more comprehensive phase 1 trials, we may be able to get what is called as EUA. This is all a new thing for regulators as well. But with this, we will definitely go into phase 2 and then phase 3 clinical trials. These trials would have will focus more on the selected dose. We have a comparator controlled. For COVID, most likely it will be just a placebo because there is no approved vaccine for COVID yet. If any new vaccines get developed, then they become comparator for future vaccine. We want to study this in all age groups as much as possible, but some of the key age groups that are requiring the vaccine, like the 60+ adults or people with underlying health conditions, they will definitely be tested in this. And while all of this is going on, we are in parallel developing the manufacturing process for these products. We have to scale up these manufacturing processes because in vaccines, the products are very complex. And we have seen cases where when we go from a small scale to large scale, the vaccine properties sometimes change. So we have to develop extremely detailed characterization of these vaccines so that when we scale up the manufacturing process, we control the vaccine properties very well so that we maintain the safety and immunogenicity of this product. And both of these tracks will end with a marketing authorization and launch of this vaccine, where the regulators will review the clinical data, manufacturing process data, and currently, the regulators are also developing many new guidelines because some of the old guidelines may not be applicable for COVID-19. So it's a very dynamic field with a lot of work happening right now, and Biologically is also participating in this pathway. We are also developing 2 types of vaccines as we speak. Let me just give you a few considerations that go into our clinical trials. The first thing, which in the past we always had a correlate of protection. So for a established product, there always a correlate of protection in terms of antibody titers, which allows us to do trials in a limited number of subjects. So if you already know what antibody titers can protect an individual from future infection, as well as a duration or durability of this protection, then that gives a great advantage in doing clinical trials because we know exactly what to hit for. But in the absence of that, which is the case currently with COVID-19, there are no correlates of protection established right now. So we hope that a lot of work which is currently underway can get to a correlate of protection very quickly, because then developing a vaccine becomes faster because you don't have to wait for a very large efficacy trials. We'll see whether we can meet that correlate of protection soon. We also rely on prior use of vaccines of similar nature, like MERS vaccine, SARS vaccines, which were tested in animals, and SARS vaccine was just tested in limited number of patients. So we hope some guidance comes out of that would allow us to assess the vaccine safety and efficacy very quickly. We are also developing a lot of key analytical assays as we enter into clinical trials, because we have to establish the neutralization of the virus either through a virus neutralization studies, which are done in vitro manner with known cell lines, or the animal challenge model that Professor Vanham showed with monkeys, rhesus monkeys. Second thing is, which is very important, is vaccine safety, especially with there is some fear of antibody-dependent enhancement of infection. And we hope the experience we observed with dengue with 4 serotypes, I hope it doesn't pan out for COVID-19, because in that case, then it introduces significant variability. We are also looking for reactogenicity because with the adjuvant, some of the adjuvants sometimes give a lot of reactogenicity. So we are looking at that as well. And one more thing which we are also worried about is what is called as Th2 immunopathology, which is something similar to the antibody-dependent enhancement. Because if that happens, then in some populations, the vaccine can actually do significant harm during the subsequent infection. So during the safety trials, we are looking at all of these markers very carefully. I'm very happy to report that WHO has initiated solidarity trials, which are vaccine trials, which will they carry out all over the world, and they will enroll a lot of manufacturers in that. We hope that they will quickly develop efficacy endpoints from some of these solidarity trials. And these are trials with multiple vaccine candidates. And we also would participate in some of these trials and make our candidate vaccine available to them. Because once you have data which is centralized, assessed in a central manner from trials which are happening in many parts of the world, I believe it will get us to a safe and efficacious vaccine much faster than individual manufacturers doing individual trials in a localized setting. So with this unprecedented global effort underway, I believe we'll be rapidly developing the vaccine. So one last aspect of any vaccine is developing a vaccine is one thing, but as you know, this is a pandemic. And so we have to make our vaccine available to millions of people. And so we need to develop a vaccine that actually is amenable for that. And the target product profile that we are working towards will address this supply challenge. So we are developing a vaccine which will be fully liquid, suitable for direct injection. Hopefully, it can be handled at room temperature and doesn't require extremely complicated cold chain distribution systems. We'll develop a multi-dose product, which again is very important in campaign settings, where we will make our product available as a 10 or 20 dose vial, which can be given in a very compact manner to many subjects. The vaccine manufacturing requires qualified infrastructure, which is essential for large-scale manufacturing. So the manufacturers in India are geared towards that. We ourselves have very large capacity to handle multiple types of antigens, as well as multiple types of formulations, as well as fill finish. We have already a proven expertise and experience to supply more than 500 million dose vaccine annually. So we already supply very large quantities of vaccines with our infrastructure. And
Professor Vanham.
Yeah, okay. But maybe Dr. Vikram can already answer the question.
Yeah, I'll repeat the question actually. See, there are a lot of claims from across the globe and from also India that the vaccine could be available in 2020 itself. But as per Professor Vanham, the earliest the vaccine would be available would be sometime in 2021. But at the same time, I'm hearing Professor Dr. Vikram talking about the process of EUA, which seems to hasten up the process, and it is likely that the vaccine can come into the market soon. So we just want to know, because everybody wants this vaccine right now, can you just tell us how early this vaccine can come into the market?
Yeah, well, I already explained my position. I think you need time for these phase 1, phase 2, 3 trials, and especially for the efficacy trial, because it depends on which target population you have, how many people you will have to include, and how long you will have to follow them up. But I don't think it is possible to do it in less than 3 to 6 months. I don't think it's possible. And then when you're sure you have a good candidate, as Dr. Vikram explained, you still have to produce this vaccine. And of course, depending on the type of the vaccine, it will take more or less time. If it's an inactivated vaccine, okay, there is a large production capacity. If it's a vectored vaccine, it will take more time to pump up these production capacities worldwide. I understand from Dr. Vikram, but also yesterday from Dr. Stoffels from Johnson & Johnson, that indeed companies are in contact with each other, large companies worldwide, and that they will support each other, that whatever candidate pops up, they will not start, that's what I promise, not start to compete, but take the best candidate on and work together to produce massive amounts. But even then, you cannot produce 1 billion doses overnight. So I really don't see, I mean, I wish, because I'm also 65, so I wish that I had a vaccine tomorrow so that I could meet my grandchildren and my family again. You see, but this is, yeah, I don't know what Dr. Vikram's view is on that.
So we definitely are in the race, and I think if the first vaccine we see good efficacy, I think it can be given to frontline workers very quickly. I believe at least it will be available to them before the end of the year. I'm hoping that the first 2-3 vaccines which are currently in development would at least get to those by end of the year. General population, we'll see, maybe slightly longer. But we have done a lot of great things in the past, so we'll try to rise to the occasion.
Okay, happy to hear that.
That's great news, actually, Vikram, from our part of the world. Actually, we hope it comes very fast. Vikram, I just understand that it's a live attenuated vaccine. Will it be effective against all strains of COVID-19? And will it be effective against the further mutations that are likely to happen in this COVID-19? It's a basic question, as a not as a clinician, but as a general public. This is what people want to know.
So a live attenuated vaccine is actually it's actually somewhat different than the COVID-19 wild type virus, because otherwise it will not be attenuated. But the key parts of the virus are kept constant in it, and so this virus is not known to mutate that much like a flu vaccine. Flu virus is much more shown mutations, so that's why you have a different vaccine every year. But in this case, this strain hasn't mutated that much. At least in the last 3 months, it's been in circulation. So the live attenuated vaccine, actually the first vaccine which has gone into trial, the vaccine from Oxford University, that is a chimeric vaccine which uses a component of the COVID-19, combines it with the chimp adenovirus, and then uses it as a vector. So it is not a classical live attenuated vaccine. It's I would call it a chimeric vaccine. So there, the spike protein part of the COVID-19 hasn't mutated that much. So we hope that at least in the short term, it should be a reasonably constant vaccine.
Yeah. So I think I want to add on that indeed it will be important that the actual viruses that circulate should be taken into account in neutralizing antibody tests, etc. And apparently the Chinese vaccine, the inactivated vaccine, they have done that. They have collected strains from over the world and shown that the antibodies they induce, that they are able to neutralize those different strains. And indeed, this virus has a proofreading enzyme, so it doesn't mutate. It corrects some of the mutations it makes. There is possibility of recombination, but it has not yet been seen, and in any case, it will not happen that often than with influenza. That is for sure. Now there have been reports already early on that there were two types, the S and the L type, I think, in China. So we'll have to see, and we'll have to follow up how this virus is going to evolve. That will be a main point of attention. Then the other point is about the attenuated vaccine. I see that there is sometimes confusion about the concept, but classically an attenuated vaccine is the same virus that has been mutated by passing in culture many times. And so it's the complete virus, but it's attenuated by passage in culture. Now, today there are molecular ways to do that, like codon de-optimization. I understand that in India, they're also involved in this process. So that would be a classical attenuated vaccine. But of course, there is always the fear that this vaccine could revert to wild type, as has happened 50 years ago with the first generation of polio vaccines that were also attenuated vaccines that reverted and that have caused a major outbreak in Germany at the time. So I think for such a dangerous virus, it is a concept that is not the first choice. But then you have this vectored vaccine, and that is different because there you have another virus that is innocent or that doesn't produce pathology, and you insert just one protein, the S protein of COVID in it, and that is much safer, of course. So just to pinpoint this concept right.
Professor Vanham, thank you for that information. We know that most of the vaccines are diverted towards the S1 subunit of the spike protein. Are there any efforts that that spike S1 subunit, whatever mutations are happening in the virus are in the S1 subunit? Are there any efforts that are being targeted S2 subunit, which is supposed to be a conserved region?
Yeah, I don't know. At this moment, everybody's looking mainly at the S1, so the receptor binding part, the receptor binding domain, because that is the point of attachment. But indeed, there are other possibilities. There is possibility, like in other virus infections, neutralizing antibodies do not only address this receptor binding domain, they sometimes also address the fusion domain, which is the S2 part of the protein. And then there is also another protein in the case of SARS-CoV-2, which might be interesting, the E protein, which is less variable and which is also a membrane protein. But we'll have to see whether that E protein in the natural infection or in models also is able to induce neutralizing antibodies. But that is a potential target as well. But it's not the first target for now. We first have to investigate the most logical target, and only if that doesn't work or is not optimal, we could look at additional alternative targets.
Yeah, thank you.
Professor Vanham, as a virologist, actually, there is a myth. People believe that, I don't know whether it's true or not, people who are vaccinated for BCG, who got BCG vaccination done, are less prone to COVID infections. What is your opinion as a virologist?
Well, I think there is no evidence. There is no evidence for that, and I do not really know where it comes from. But BCG has been used for a number of things as an immunostimulant, and for instance, it has been used in bladder cancer as an immunotherapy. So BCG is a kind of one of these magic bullets. Of course, it induces a very strong Th1 response, but I don't see how this would protect us really against this particular virus. Of course, BCG could be used as a vector as well. BCG, you could insert the S protein in BCG. That is a possibility, but I don't know whether that kind of effort is ongoing. The BCG in itself, honestly, I don't see how it could protect us specifically against this infection.
So actually there is a trial going on in India with a recombinant BCG vaccine, which will be given to healthcare workers and some frontline workers. So that trial is starting soon in India.
Yeah.
So some audience request is to know what is herd immunity and how much time it will take to develop herd immunity in the populations.
Yeah. Well, of course, herd immunity is a concept. It's a theoretical concept. You can only have herd immunity, of course, if you have long-standing neutralizing antibodies. If a virus or an other infectious agent induces neutralizing antibodies that are there to stay for a while, right? Because not everybody gets infected at the same time, and so on. So what we still have to, in Europe, the situation now is, as you know, Europe has been hard hit by this epidemic, and the first seroprevalence studies are upcoming now. So even in the hardest hit countries, it seems that less than 10 or even less than 5% of the population is seropositive at this moment. So clearly more than 90% has no antibodies. So even after this, really vigorous epidemic, there is certainly no herd immunity in the European population at this moment. And the same is true, of course, for China, where the epidemic has also been put under control. So you would need, according to various models, you would need at least 70% of the population that has neutralizing antibodies. So this is a very difficult concept, because then you need to let the virus go and infect essentially everybody. And then there is a lot of uncertainty how long these antibodies, once induced by natural infection, will last, because in other coronaviruses, they last for 1 year, 2 years maximum. And also with SARS, there was a decline with the people that were infected with SARS. There was a clear decline in antibody levels, and even for this virus now, we have already seen that neutralizing antibodies in some of our patients have declined soon after infection. So if you take all that evidence together, well, it's preliminary evidence, but if you take it all together, then herd immunity is not for the coming year or so. You would need much more time, and so I think we cannot wait for herd immunity. We need a vaccine that is better than the natural infection.
So it's going to take a long time before the herd immunity develops in the populations.
Yeah. Of course, it will ultimately develop if you let the virus go. That will happen, because more and more people will be infected, and those that are very susceptible will die. Yeah. That's how it works in nature. But I don't think we are prepared to let that go.
So somebody is questioning whether this pandemic is going to remain, or is it going to become endemic in different areas?
My opinion, I don't know. My opinion is that this virus is clearly here to stay, and that it will be with us as long as we don't eliminate it. And of course, the question now is how fast will it spread into the more tropical areas of the world. And of course, I'm also at the Institute of Tropical Medicine, so we have several people in Africa and in South America, etc., watching the epidemic. And at this moment, it doesn't seem that it's spreading so rapidly as it has spread in Europe and in the United States. So I've read a paper also yesterday about the structure of the S protein, that because of the flexibility and because of certain physicochemical characteristics, it would indeed be sensitive to temperature, and so but we'll see whether really this virus is going to have a seasonal periodicity in the northern hemisphere, and will be spreading at a much more lower level in warmer countries. It's something we will learn in the coming months.
Thank you so much, Professor Vanham and Dr. Vikram, for the beautiful updates actually you've given us. Thank you so much for sparing your valuable time.
Thank you.
We hope, all of us hope, as Dr. And we all wish Dr. Vikram that he comes up with a vaccine very soon into the market, and hope we all start using it. Thank you so much again, and we are signing off from AIG Hospitals, Hyderabad, India. Thank you so much once again for your valuable time.
It was a pleasure. Thank you very much.
Thank you.
Thank you.
Bye.
Bye.